Every cell in your body contains something that is not you, not built from your parents' DNA, not part of your evolutionary lineage in the way your hands or your liver are. These structures have their own genome, a separate circular strand of DNA completely unlike the linear chromosomes in your cell's nucleus. They replicate on their own schedule. They divide independently of the cell that houses them. And they have been living inside cells like yours for approximately 1.45 billion years, long before the first animal ever existed, long before complex multicellular life had invented itself, in an ocean that looked nothing like the planet you are standing on now. They're called mitochondria, and they are the reason you are alive right now. There are roughly 37 trillion cells in your body. Most of them, all but your red blood cells and a handful of specialized cell types, contain between 1,000 and 2,000 mitochondria each. In your heart muscle cells, where the energy demand is constant and relentless, that number climbs to 5,000 per cell. In the neurons of your prefrontal cortex, mitochondria account for up to 25% of the total cell volume. In total, the combined mass of mitochondria in your body accounts for roughly 10% of your body weight. You are carrying approximately 7 to 8 kilograms of these structures right now, and without them, every cell in your body would exhaust its usable energy reserves within seconds and die. The way they produce that energy is one of the most extraordinary mechanical processes in all of biology. And the fact that they remember, not in the way a brain remembers, but in a deeper, more ancient way, is a story the science is only beginning to tell. That second part is the part that should concern you personally. But to get there, you first have to understand what they are and where they came from. And that story begins not in medicine or physiology, but in planetary geology. To understand mitochondria, you have to go back to a moment roughly 1.45 billion years ago that changed the trajectory of all complex life on Earth. It happened in a single cell. No one witnessed it. No fossils recorded the exact interaction. But the evidence that it occurred is sitting inside your chest right now, running at several thousand revolutions per minute, never pausing. Here is the theory as it stands today, supported by genomic, ultrastructural, and biochemical evidence accumulated across 6 decades of research. An ancient archaeal cell, a member of the domain of life older than bacteria and distinct from them, engulfed a free-living alpha proteobacterium. Engulfment is not unusual. Cells consume other cells. But in this particular case, the bacterium was not digested. It survived inside its host. The host gained access to a far more efficient method of extracting energy from organic molecules. The bacterium gained shelter and a stable chemical environment. Over hundreds of millions of years, the two organisms became so metabolically intertwined that neither could survive without the other. The bacterium gradually transferred most of its genes into the host cell's nucleus. It lost its outer cell wall. It became optimized for a single function, producing energy for the host. And it never left. That captured bacterium is your mitochondria. You are the host cell. The relationship is 1.45 billion years old. The specific identity of the archaeal host has been one of the most contested questions in evolutionary biology. In 2015, Thijs Ettema and his colleagues at Uppsala University in Sweden published the discovery of a new archaeal phylum they named Asgard archaea. These were retrieved from sediment samples taken from hydrothermal vents near Loki's castle in the North Atlantic Ocean. Genomic analysis revealed that the Asgard archaea possessed genes previously thought to be exclusive to eukaryotes, the domain that includes all cells with nuclei, including yours. The discovery placed the Asgard archaea as the closest living relatives to the ancestral archaeal host that eventually gave rise to all eukaryotic life. The paper was published in Nature and immediately reframed the debate about eukaryotic origins that had been running since the 1970s. This means the line of descent that leads to your mitochondria begins with a bacterial cell in an ancient ocean, captured by an archaeal ancestor related to organisms still living today in deep-sea sediment near a geological feature named after a location in Norse mythology. The scientist who spent most of her professional life arguing for the bacterial origin of mitochondria was Lynn Margulis, a biologist at the University of Massachusetts Amherst. In 1967, she submitted a paper titled On the Origin of Mitosing Cells to 15 scientific journals. All 15 rejected it. The consensus at the time was that mitochondria had evolved within cells from the cell's own internal structures, that they were differentiated components of the cell's own machinery. The idea that they were captured and domesticated bacteria was considered unserious. When her paper was finally accepted and published in the Journal of Theoretical Biology, the reception from mainstream cell biology ranged from skeptical to dismissive. Then molecular biology developed the tools to test it. The mitochondrial genome was sequenced and found to be unmistakably bacterial in structure and origin. Circular, compact, with gene organization matching that of alpha proteobacteria. Mitochondria were found to reproduce by binary fission, the same process bacteria use, not by the coordinated cell division process eukaryotic cells use. Their ribosomes turned out to be bacterial in size and antibiotic sensitivity. Chloramphenicol, an antibiotic that kills bacteria by blocking their ribosomes, also blocks mitochondrial protein synthesis. It has no effect on the cytoplasmic ribosomes in the rest of the cell. By the 1990s, Lynn Margulis' endosymbiotic theory was textbook consensus. She had been right for more than 20 years before the field caught up with her. That history matters because it illustrates something important about how deeply embedded the bacterial origin of mitochondria actually is. It is visible in the antibiotic sensitivity profile, in the genome structure, in the ribosome size, in the reproduction mechanism, in the membrane lipid composition. And here's what that bacterial origin means for the structure you carry in every cell. Mitochondria have their own DNA, not a copy of the DNA in your nucleus. Genuinely separate, genuinely their own. Human mitochondrial DNA is a circular molecule of approximately 16,569 base pairs, encoding 37 genes. The nuclear genome contains roughly 3.2 billion base pairs encoding approximately 20,000 genes. But those 37 mitochondrial genes encode something irreplaceable: the core protein subunits of the electron transport chain, the machinery that actually synthesizes ATP, adenosine triphosphate, the molecule your body uses as its universal energy currency. The mechanism is worth understanding in physical detail because it is unlike any other biological process, and because it is happening in your cells without pause right now. Along the inner mitochondrial membrane, a membrane folded into deep ridges called cristae that increase surface area by a factor of 5 compared to an unfolded sphere. There are 4 large protein complexes embedded side by side. Together they constitute the electron transport chain. Electrons stripped from food molecules, from glucose broken down in glycolysis, from fatty acids processed in beta oxidation, from amino acids redirected through the citric acid cycle, are passed sequentially from complex 1 to complex 2 to complex 3 to complex IV, losing energy at each handoff. That energy is not released as heat. It is captured and used to do mechanical work, pumping hydrogen ions, protons, from the innermost compartment of the mitochondria, called the matrix, through the membrane into the intermembrane space. This creates 2 simultaneous gradients across the inner membrane: a concentration gradient, because proton density is higher outside the matrix, and an electrical gradient, because protons carry positive charge. Together, these constitute the proton motive force, a form of stored electrochemical potential energy that sits at roughly 180 to 200 millivolts across a membrane less than 8 nanometers thick. The electrical field this represents is approximately 30 million volts per meter. By comparison, the electrical field strength that will arc lightning through dry air is about 3 million volts per meter. The inner mitochondrial membrane sustains 10 times that field strength, continuously, in every one of your living cells. That field drives the 5th complex, ATP synthase. ATP synthase is a rotary molecular motor, not a metaphor. A physical rotor with a shaft, a stator ring, and a rotating C subunit ring that spins physically as protons flow back through it down their electrochemical gradient. The C ring in human ATP synthase contains 8 C subunits, and each complete rotation, requiring 8 protons to pass through, produces 3 molecules of ATP. At peak activity, the rotor spins at 100 to 150 revolutions per second. The machine was given the name Fo-F1 ATP synthase by Paul Boyer and John Walker, who worked out the rotary mechanism and shared the Nobel Prize in Chemistry for it in 1997. Boyer's contribution was the binding change mechanism, the insight that the rotation was not a consequence of ATP synthesis, but the cause of it, with physical rotation mechanically deforming the enzyme's active site to force the synthesis of each ATP molecule. When he first proposed it, it was considered implausible. Electron microscopy eventually revealed the rotating structure directly. The physicist in the room would note that this is structurally identical in principle to a hydroelectric turbine. Water pressure stored behind a dam drives rotation, and rotation drives a generator. Proton pressure stored across the inner membrane drives rotation, and rotation drives ATP synthesis. Your mitochondria are running a biological power station at the scale of nanometers, and they have been doing it continuously since before your first cell divided. But there is a second layer of this story that the energy mechanism alone does not contain. And this is the layer that connects you specifically, your history, your mother's history, your life choices, to something 1.45 billion years old. Mitochondrial DNA is inherited almost exclusively through the maternal line. When a sperm fertilizes an egg, the sperm's mitochondria are tagged almost immediately for destruction. Ubiquitin, a protein the cell uses to mark structures for degradation, is attached to paternal mitochondria within hours of fertilization, and they are eliminated through autophagy before the embryo's first few divisions are complete. The mitochondria in every cell of your body came from your mother. Hers came from her mother. The chain is unbroken from the first cell of your embryo back through human history. Because mitochondrial DNA does not undergo the recombination that shuffles nuclear DNA each generation, mutations accumulate through a clean, linear history of point substitutions. Geneticists can use this property as a molecular clock. By comparing mitochondrial DNA sequences across humans worldwide and counting divergence, they can estimate how long ago any two people shared a common female ancestor on the purely maternal line. This is how the concept of Mitochondrial Eve was established by Rebecca Cann, Mark Stoneking, and Alan Wilson at the University of California, Berkeley in 1987. Their analysis placed the common matrilineal ancestor of all living humans approximately 150,000 to 200,000 years ago in sub-Saharan Africa. Current estimates from the most comprehensive datasets place her at approximately 150,000 years ago. The mitochondria in your cells carry an unbroken chain of maternal descent reaching back to a single woman. The genome inside them is a continuous record of that lineage. Every mutation accumulated in that 150,000-year chain is present in the mitochondrial DNA you carry, readable by sequencing technology that costs less today than a month of internet service. And now here is the layer that is genuinely new and genuinely contested, which means it should be described carefully. In the last decade, research has established that mitochondrial DNA can be epigenetically modified and that the expression of mitochondrial genes can be altered by environmental stress, metabolic states, and developmental signals. Epigenetics refers broadly to modifications that change how genes are expressed without changing the underlying DNA sequence. The addition of methyl groups to DNA, the modification of the protein spools that DNA wraps around, and related mechanisms. The evidence for transgenerational mitochondrial epigenetic inheritance is preliminary and hotly debated. But the mechanistic basis has been demonstrated in pieces. Mitochondrial DNA methylation, the addition of methyl groups directly to mitochondrial DNA bases, has been documented in humans and in several other species. The enzymes capable of methylating mitochondrial DNA have been identified. And in Caenorhabditis elegans, the nematode worm, whose short generation time makes multi-generational studies feasible, work from the laboratory of Johan Auwerx at the École Polytechnique Fédérale de Lausanne has shown that mitochondrial stress responses in one generation produce heritable changes in gene expression in subsequent generations, transmitted specifically through the maternal line. What this research points toward, carefully, because the human evidence is not yet definitive, is that your mitochondria are sensitive to your life history in a way that may not stop with you. Caloric restriction, sustained aerobic exercise, cold exposure, and intermittent fasting all produce documented changes in mitochondrial gene expression, mitochondrial morphology, and mitochondrial population density. They activate a process called mitophagy, in which damaged or dysfunctional mitochondria are tagged and selectively destroyed. The mitochondria that survive are the ones that perform best under stress. This is the cellular equivalent of natural selection, compressed into a single organism's lifetime. The interventions that drive these changes have been measured at the molecular level. Kirk Erickson at the University of Pittsburgh demonstrated in 2011 that sustained aerobic exercise over 1 year produced measurable volumetric increases in the hippocampus of older adults, measured by citrate synthase activity, a biochemical proxy for mitochondrial volume. Aerobic training in previously sedentary adults increases mitochondrial density in skeletal muscle by 40 to 50% over 8 to 12 weeks. David Sinclair at Harvard Medical School demonstrated in 2013 that raising cellular NAD+ levels in aged mice produced muscle mitochondria morphologically and functionally indistinguishable from those of young mice. Within 1 week of treatment, the animals were not younger. Their mitochondria were behaving as if they were. Modern life, by contrast, disrupts mitochondrial function through at least 3 well-characterized pathways. High-fructose diets drive excess production of reactive oxygen species at complex 1 and complex 3 of the electron transport chain, damaging mitochondrial membranes and mitochondrial DNA faster than repair mechanisms can compensate. Chronic sedentary behavior reduces metabolic demand, and without demand, the signal for mitochondrial biogenesis, primarily mediated through PGC-1A, the master switch for mitochondrial production, is not triggered. Artificial light at night suppresses melatonin, which has been documented to function as a mitochondria-targeted antioxidant in multiple cell types, reducing protection against oxidative damage during the hours when mitochondrial repair is most active. These are not speculative connections. They are documented mechanisms with identified molecular intermediaries. There is one final layer. The outer membrane of your mitochondria retains molecular signatures of its bacterial origin. Chief among them is cardiolipin, a phospholipid found almost exclusively in bacterial cell membranes and in the inner mitochondrial membrane of eukaryotic cells. It is an evolutionary fingerprint of the original bacterium that was captured 1.45 billion years ago. When mitochondria are severely damaged, when cardiolipin escapes into the cytoplasm, or when mitochondrial DNA leaks out of the organelle, the immune system responds as if detecting a bacterial invasion. Because from the perspective of the pattern recognition receptors on your immune cells, that is exactly what the molecular signature looks like. The danger signal for mitochondrial damage and the danger signal for bacterial infection are structurally the same signal. This is not a design flaw. It is a trace fossil of the original merger event. The immune system has not fully updated its recognition protocol in 1.45 billion years, because from a functional standpoint, there has been no reason to. Right now, in every cell of your body that contains mitochondria, in your heart, your neurons, your liver, your muscle fibers, the electron transport chain is running. The proton gradient is being maintained. The ATP synthase rotors are turning at more than 100 revolutions per second. The system has not paused since before you were born. It did not pause in your mother before you, or in hers. The chain of uninterrupted mitochondrial activity reaches backward through your entire maternal lineage to the woman in sub-Saharan Africa 150,000 years ago, and from there through billions of years of cellular life in seas that predate all animals, all plants, and all complex organisms. You thought of mitochondria, if you thought of them at all, as the powerhouse of the cell, a label from a textbook diagram, a fact memorized and forgotten. But the powerhouse is a captured organism. It has its own genome, its own evolutionary history, its own quality control processes, and its own record of your metabolic life. It responds to what you do. It is changed by how you live. And the science suggests it may pass some of what it has learned to those who come after you. through the one inheritance pathway that runs from mother to child without interruption. You are not a single organism. You have never been a single organism. The bacteria never left. They just became essential.